succinate was practically gone, as was the acetate. So something about room air had disabled the fermentation pathways of B theta. It had interfered with the bug's ability to generate energy. So when they went and looked and pieced together the pathways here, they found there are two key enzymes that are disabled by oxygen. One is fumarase, and the other is pyruvate ferredoxin oxidoreductase. They're inactivated because they contain iron sulfur clusters. And what they specifically show is that fumarase is inactivated by superoxide. But the big finding is that P4 was actually poisoned by molecular oxygen itself and not the aflatoxin species. So that's a rare thing. And so from this they concluded that one of the reasons that B theta might not grow in the presence of oxygen is because it has enzymes that are specifically poisoned by molecular oxygen itself. So this is the framework in which we were thinking about bee strategy. Although this came later, once we had figured out what might be going on. First we needed to get lucky, and we did. When I was a rotation student in the lab, I was working on a project that I don't even remember what it was, but I was working under anaerobic conditions. So the chambers got a little bit of oxygen in there. We didn't have a meter, so you had to go in every once in a while and do this cumbersome assay to figure out how much oxygen was in it. So we didn't do it very much. And one day I played with wild-type Bacteroides, and I got very few colonies that came up. In fact, they're about 1,000,000-fold less than they should have been. And I went to Tony at the time, and I showed him, and we both kind of scratched our head, and I think we both realized that I screwed something up. Which is probably true. But what he was able to find out, I went on to my next rotation, and went in and checked the O2 concentration in the chamber, and it crept up significantly. Now it was instead of being at 0.05%, it had crept into what we call the microaerobic range of 0.2% up to 1.5%. He found that he could purify these colonies under those microaerobic conditions. So now they were growing under 1.5% oxygen, which represented a 30-fold increase in the amount of O2 under which they could grow. We didn't know what to make of those. We couldn't make heads or tails, but they seemed like they might be mutants. So what Tony asked was, could you isolate what we were calling oxygen-enabled mutants from different background strains that had mutations in the oxidative stress response? So for instance, you might hypothesize that by having a mutation in superoxide dismutase, that strain can't give rise to oxygen-enabled colonies because it's being poisoned. That wasn't the case at all. You could knock out cytochrome oxidase, catalase, superoxide dismutase, and they would all give rise to colonies under anaerobic conditions with frequencies similar to wild type. He also made a disruption in a operon that our lab has shown is important for Bacteroides aerotolerance, and again, same frequency as wild type. So we asked the opposite question. Could you get a mutant strain that now could give rise to more oxygen-enabled colonies? Some of the oxidative stress response of B. fragilis is controlled by the transcriptional factor OxyR. It binds in the promoter region and is usually inactive, but in the presence of reactive oxygen species, there's a conformational shift. It makes contact with RNA polymerase and drives expression of downstream genes. The Smith lab at East Carolina University had isolated a strain that encoded a constitutively active OxyR. So in other words, the oxidative stress response is constantly turned on in this organism. So when you play to that, you might hypothesize that it could give rise to more oxygen-enabled colonies, but that was not the case. Again, it was just like wild type. We had another stroke of luck in that Tony had happened to make one other mutant. He had knocked out a gene called rubidoxin oxygen oxidoreductase, or RU. And as the name implies, it can reduce oxygen, and it does so all the way to water. So it's a defense mechanism. And it had been studied in sulfovibrio, which is another anaerobe. They found when they knocked out RU that these bugs were more sensitive to 1% oxygen. So again, you might hypothesize, by knocking out RU, perhaps we were not able to get any oxygen-enabled colonies to come up, and of course it played with 100% efficiency. Meaning, we got as many colonies that came up microaerobically as we did anaerobically, which is the exact opposite of what you would have thought. So we look at what RU is, where it is on the genome. That was quick. And thank you.